So this begins a new chapter in your book. Um, it also really begins where we start thinking critically about these pathways. Okay, and glycolysis is a perfect place to start because it's number one, it is for essentially every organism, it is the primary way to make energy, at least part of the primary way to make energy, right? Every organism has a way of converting glucose or a related sugar into pyruvate. Now, I will say that there are some organisms that do not have a fully functional glycolysis, okay, which is interesting. But they do have mechanisms to bypass the places that they lack the genes to do the certain reactions, okay? Um, and the idea here is that we are starting with glucose, and ultimately we're breaking it down into pyruvate, right? And during this process of making pyruvate, we are also consuming some of the oxidized form of NAD+, and we're generating some NADH, which means during this process, we are oxidizing the carbons of glucose, at least to some degree, okay? And then once we make pyruvate, then the fate of the pyruvate will depend on what's going on in the cell. If the cell has plenty of oxygen around, then it can then enter into aerobic respiration, and where the pyruvate is then oxidized the rest of the way down to carbon dioxide. Okay? If there's not enough oxygen around, then the problem becomes that once we stop a pyruvate, we've made some energy, we've made some ATP, so that's great. It's a way of making ATP for cells. Not amazing, but it does it. But the problem is that the free NAD plus pool that we've generated has been depleted because as glycolysis proceeds, it consumes NADH, or it consumes NAD plus and produces NADH. But for this process to be able to continue, we need to be able to regenerate the NAD plus. And that's where the idea of aerobic or anaerobic respiration comes in, right? If there's not oxygen around, so the aerobic pathways can be active, we have to have a way of regenerating our NAD plus. That's what anaerobic respiration is. And for us, for mammals, for eukaryotes, for most eukaryotes, that boils down to converting pyruvate to lactic acid. That process of converting pyruvate to lactic acid will then oxidize the NADH back to NAD+, and glycolysis can continue in a catalytic way. Okay. Other forms of anaerobic respiration, as you probably know, is ethanolic, and that's the one that makes delicious things. Okay, so glucose catabolism. It's okay to laugh at my stupid jokes, y'all. It's fine. I'm hilarious. And that's what you laugh at. Thank you. Okay, so glucose catabolism. Remember, catabolism is breaking big molecules down to make energy. Okay, it starts with dietary sugars. Whether you are a mammal or you are a bacteria, at the end of it really boils down to what you can bring into your cell. Okay, for us, most of the sugars that we eat are not simple sugars, right? They're not single hexoses. They are more complex sugars. So we have enzymes in our saliva, in our pancreas, in our digestive system that will break them down into the simple monosaccharides that we can then move into our cells. One way or another, this gets converted into glucose or something similar to glucose. Okay, and then this can enter into glycolysis. Glycolysis is what we're really studying today, which breaks it down to pyruvate. From there, it can then be fermented into ethanol or lactate, depending on what organism it is, if there's no oxygen around. If there's plenty of oxygen, then we can oxidize glucose the rest of the way to carbon dioxide and water and produce robust amounts of ATP. Okay, but so this question down here, why did evolution choose glucose, is kind of an interesting one, right? And you can come up with lots and lots of different answers, but the three that really continue to emerge, no matter how you look at it, is that glucose is one of the only sugars that if you simulate prebiotic conditions, it's a stable sugar. It's stable enough that it will be made in these primitive biochemical systems. Okay, so it could be made in a really, really potent reducing atmosphere. Okay, so that's one reason, probably because it was already there, it was around. Another one is it's the most stable hexose, right? If you think back to organic, and I know none of us want to think back to organic, except maybe Kenny, because he likes it, and that's why he's weird, right? But remember, 6-carbon, like so, 6-carbon molecules, 6-carbon rings adopt either a chair or a boat conformation, and the boat or the chair conformation is more stable, and when the bigger functional groups are in the equatorial position, that's when things are more stable. And if you look at the structure of glucose, all of the alcohol groups, all of the larger functional groups are in the equatorial position, making it the most stable of the hexoses. Okay, so life chose it because it's the most stable. Okay, and because it's really stable in its ring form, and it's not as likely to then transition into its linear form, which makes it less likely to randomly glycosylate proteins or other molecules. Okay, and we won't really get into that this semester, but that's one of the reasons that it's really important, at least for more complex organisms. Okay, so if we think about glucose metabolism in general, right? So this energy, so this diagram kind of shows some of the things that can happen, and really most of the things that can happen. Okay, so the thing we're going to focus on today and Thursday is that glucose can be oxidized to make pyruvate, ultimately to make energy. Okay, under conditions where there's plenty of energy around, glucose can be stored. If you're humans, or mammals, it's stored in the form of glycogen, right? We have glycogen granules in our muscle tissue and in our liver. So it's a storage form of energy. If you are a plant, it can be stored in the form of starch. Okay. Glucose is exceptionally important in making nucleic acids. So it can go through this alternate pathway called the pentose phosphate pathway that creates ribose 5-phosphate. Okay. This is the only way to make NADPH. It's the only way to make pentoses, including ribose, which ultimately helps us make nucleic acids and other cofactors. It's also an important structural component of cell walls and some proteins that decorate cells, right? So lots of fates. We will think about 3 of these fates this semester. We'll think about oxidation, we'll think about storage, and we'll think about the pentose phosphate pathway. All right, so y'all have all probably heard this before, right? There's 2 stages of glycolysis. There's the investment phase and there's the payoff phase, okay? The idea with the investment stage is that we are consuming ATP to prepare glucose to make energy for us, right? We have to prepare it to be able to produce energy. And for this, we have to invest some energy first, okay? So in stage 1, we invest some ATP, ultimately converting it to fructose 1,6-bisphosphate, which is then split in 2. Once it's split, it enters into our payoff phase, where 2 equivalents of 3 carbon molecules are moved through these oxidative and transformative steps that ultimately produce 4 ATPs and give us 2 equivalents of pyruvate. Okay, so I'm confident that you have thought through this before at some point in one of your other biology courses. Okay, so at this point, we'll go ahead and take a break. I'll give you some time to work through the first couple questions on today's activity, which introduces some of the themes we're going to think about for glycolysis.